The eyes of the world today stunned by this image. This was debris from a meteor racing toward Earth, streaking across the sky there, crashing into the ground in those woods.
A meteorite that crashed to Earth has revealed something scientists never thought possible. When it was cracked open in a lab, scientists didn't just find rock and dust. They uncovered strands of DNA unlike anything ever seen before. The DNA structure doesn't match humans, animals, or even known microbes. This discovery has shaken science to its core, suggesting life may not have begun here at all. Could this be evidence of life from beyond Earth? Join us as we find out how this affects the definition of life as we know it. Life in another form. It all began with a fall from the sky, a flash that crossed the upper atmosphere, and a small impact site recorded in a remote desert region. Researchers were alerted when instruments that track space debris registered the strike. A recovery team moved out, not wanting the meteorite to sit exposed for long. The rock was collected using sterilized tools and placed in a sealed container. No direct human contact was allowed. Scientists followed strict procedures because they knew that the value of any meteorite lay in its interior. In the laboratory, the meteorite was placed in a controlled clean room. Air was filtered, surfaces were scrubbed, and protective suits were mandatory. The outer layer of the rock was carefully removed, since it could have been affected during its passage through the atmosphere. What mattered most was what lay inside, preserved from Earth's environment for millions of years. Finally, when scientists cut deep into the meteorite, they found structures that looked unusual. Under high-powered microscopes, small patterns appeared inside fine cracks of the stone. These samples were placed into sealed slides and examined using spectroscopy and electron imaging. The early readings were not what the team expected. Alongside metallic compounds and silicate structures, traces of organic molecules were detected. This was not rare in meteorites. Previous finds had revealed amino acids, simple sugars, and carbon chains. But the concentration in this rock seemed higher than normal. The next stage of testing involved separating the organic material. Using chemical extraction methods, the scientists removed tiny fragments from within the rock and placed them into sequencing machines. They wanted to know if the patterns were random chains or if they showed organization, the kind of structure normally linked to life. What appeared on the screen was surprising to the entire team. The material showed repeating sequences that resembled strands of DNA. The reaction in the room was mixed. Some believed it was a false signal, perhaps the result of contamination. Others argued that the procedures were too strict for that. Every instrument had been sterilized, every surface treated, and the samples had been removed only from deep inside the rock. To rule out errors, the tests were repeated. Multiple labs were invited to conduct blind analyses on separate samples. When the results were out, they all came back the same. Inside the meteorite were long, complex molecules carrying information in a structured sequence. They were not fragments from human handling, nor did they match known plant or microbial DNA cataloged on Earth. After the discovery, internal reports circulated first, moving between specialists in genetics, astrobiology, and planetary science. Teams worked independently to verify results. High-resolution imaging was used to visualize the molecular structures, and isotope analysis confirmed that the material had been locked inside the rock for millions of years. This ruled out the chance that the DNA-like material had been introduced after the meteorite's arrival on Earth. The meteorite itself was dated using radiometric methods, placing its origin far back in the history of the solar system. Some evidence suggested it came from the outer regions, where icy bodies and primitive material have remained preserved since the earliest times. If the organic molecules inside were truly structured DNA, then they had survived an unimaginable journey through space, shielded by the rock's mineral shell. Next, geneticists decided to compare the sequences against every known database. They found no match with humans, animals, plants, fungi, or bacteria. The sequences carried their own code, not random, but ordered, as if written by an unknown biology. The data was stored securely and checked again. Each test reinforced the same conclusion. A rock that had traveled across space contained a molecular system that looked and behaved like DNA, yet it was something strange, something that did not belong to this planet. Word soon spread through research circles. A select few were invited to join the investigation, Though strict confidentiality agreements were in place, the scientists involved knew that such a claim, if released too early, could damage reputations and create public confusion. Teams worked long hours, comparing, questioning, and checking every possibility. They realized that they were looking at information that had come from beyond Earth, and that what lay inside the meteorite was far more than stone. The nature of the DNA. Once the meteorite samples were secured, the real challenge began. The research teams had to understand the structure of the strange sequences found inside. It was a signal written in molecular form, carried across space, and preserved through fire. It was not enough to say the material looked like DNA. They had to examine every detail, every base, every connection, and see how it compared to what was known on Earth. In a genetics facility built for high-security work, the extracted material was placed into advanced sequencing machines. These machines could read strands at a molecular level, producing long maps of letters that represented the chemical bases. The scientists expected to find similarities with common Earth life. Instead, the first sequences came back showing patterns that were highly irregular. The material had 4 repeating bases, just as Earth DNA does, but their arrangement was not the same. Some chains were far longer than those found in terrestrial organisms. Others contained repeating blocks that had never been documented. It was as if the code followed its own rules, forming a language separate from the one familiar to biology on this planet. As more sections were sequenced, researchers noticed unusual binding points. Earth DNA coils into the familiar double helix, stabilized by hydrogen bonds. The extraterrestrial material showed twists that formed triple-stranded segments in certain regions. This was not theoretical. It appeared again and again in independent samples. The data suggested a structure that could fold differently, creating stability under extreme conditions, perhaps even in the vacuum of space. Another surprising detail was the resilience of the molecules. When Earth DNA is exposed to radiation or intense heat, it quickly breaks down. Yet these strands remained intact despite having traveled across cosmic distances. Chemical tests revealed unique protective coatings around the molecules, as if an outer shield had formed naturally to preserve them. This was something Earth life had never developed. Teams of computational biologists were called to model the sequences. They used supercomputers to simulate how the alien DNA folded, how it might replicate, and whether it could code for proteins. The results were fascinating. The sequences contained open reading frames, regions that could, in theory, produce functional proteins. But the order of the bases was so unfamiliar that the team could not match them to any known protein families. It was a code that pointed to a system of life unlike anything studied before. Microbiologists argued over whether this material had once belonged to an organism. Some suggested it might have been part of a microbe, frozen in the meteorite long before reaching Earth. Others believed it was something more primitive, a pre-cellular form of information storage, preserved from a time before complex life evolved anywhere. To test stability, portions of the sequences were copied using polymerase chain reaction methods. This allowed scientists to amplify fragments and study them more closely. To their surprise, the amplification worked. The alien strands behaved as if they were natural templates, accepting replication processes normally used on Earth DNA. This confirmed that the material was not random. It carried a consistent, repeatable code. The differences, however, remain striking. Earth organisms share a common genetic alphabet, with codons that instruct cells to build proteins. The extraterrestrial material used codons that did not match this system. When researchers tried to interpret the sequences using the standard genetic code, the results came back nonsensical. It was as though the meteorite carried a different biological software, one that followed its own operating rules. Chemical analysis also showed a deviation in the sugars that form the backbone of the strands. Earth DNA uses deoxyribose, a sugar molecule with a specific arrangement of atoms. The alien material had a variant sugar, similar but not identical. This small change gave the molecule greater resistance to breakdown, strengthening the structure against time and radiation. Samples were also tested under extreme conditions to see how they responded. When heated to temperatures that would normally destroy Earth DNA, The strands held their shape. When exposed to ultraviolet radiation, they showed remarkable resistance. Even acidic environments did little damage. This suggested that whatever organism or system this DNA once belonged to, it was adapted to survive in places far harsher than Earth. Comparisons with databases confirmed the uniqueness. Billions of sequences from bacteria, plants, animals, and viruses were checked against the Meteorite code. No overlap was found. Not even the simplest Earth microbes showed a relationship. The alien material was entirely distinct. It carried order, complexity, and resilience beyond anything seen in terrestrial life. The longer the researchers studied, the more questions appeared. Some suggested the triple-stranded sections could store additional layers of information, a kind of built-in redundancy. Others wondered if the protective coatings meant this DNA had been designed to travel, spreading across environments until conditions allowed it to activate. What was clear, however, was that the meteorite's DNA was not a simple chemical accident. It was a message that spoke of possibilities far wider than the biology of this planet. Varying scientific reactions to the new discovery. When the first reports about the strange sequences began to circulate among scientific circles, the reactions were divided. In one genetics institute, a senior researcher paused over the data and admitted it was unlike anything he had seen in 30 years of work. Others, however, were quick to raise doubts. Extraordinary claims demanded extraordinary proof, and this was the kind of finding that could disrupt entire fields of study. Committees were set up to review the raw data. Teams of specialists were asked to repeat the sequencing independently, using samples that had been split and shipped under strict security. These labs confirmed that the structures were real and that they did not match the genetic signatures of known Earth organisms. Still, the results sparked heated debate. Some scientists welcomed the idea of an extraterrestrial genetic system. Others argued it was far too early to draw such conclusions. At conferences, sessions that normally attracted small groups suddenly filled large halls. Slides showed molecular maps, structural diagrams, and unusual folding patterns. It proved how the foundation of Earth biology could also exist in such a foreign form. The scientists who first made the discovery opened their data sets, allowing controlled access to sequence files. This level of transparency was rare in competitive fields, but it was the only way to build trust. The responses from different scientific disciplines varied. Astrobiologists were perhaps the most excited. For decades, they had argued that life could exist elsewhere, but they lacked physical evidence. To them, the discovery provided the first real material to study. Microbiologists, on the other hand, were cautious. They worried about contamination, about unknown microbes entering Earth's environment, and about the possibility of overinterpreting what might only be unusual chemistry. In genetics labs, younger researchers found themselves working on projects that felt historic. Graduate students stayed late, comparing notes, rechecking base sequences, and running computer simulations. Many described the atmosphere as tense but electric, a mixture of pressure and wonder. Meanwhile, senior scientists warned them to remain careful. If the findings collapsed under scrutiny, careers could be damaged. The scientists knew they could not control the story for long, yet they wanted to be certain before anything went public. Government agencies also became involved. Research funding bodies demanded updates. Space agencies asked for detailed reports since the discovery could affect future missions. In some meetings, officials raised concerns about security, about how such material should be handled, and whether there could be risks from studying it outside high-containment facilities. The atmosphere in academic communities was unlike anything seen in recent years. Some called it a turning point, others a distraction. Arguments broke out in private discussions and public debates. Was this truly evidence of extraterrestrial life, or simply an unknown branch of chemistry that looks just like genetic structures? The lack of consensus fueled more discussion. Rumors spread online carrying exaggerated claims about alien organisms. This only increased the need for clarity. Even among those who accepted the findings, interpretations differed. Some argued the sequences proved that life had multiple origins, that biology might arise wherever the right conditions existed. Others believed the material could support panspermia theories, the idea that life spreads across planets by traveling inside rocks and comets. A smaller group warned that it might not represent life at all, but only a chemical system that resembled DNA without ever forming living cells. If DNA could take on unfamiliar forms, what does it mean for life beyond Earth? Did life have to be defined by Earth's rules, or was it broader than that? Even with all that had been studied, that one discovery needed years of careful work before altering the foundation of knowledge. Still, the sense of a shift was clear. There's now the possibility of studying something beyond Earth for the first time. The reactions were human: caution, pride, opposition, and wonder, all playing out in real time as the evidence continued to build. Implications for life beyond Earth. The discovery of unfamiliar DNA sequences inside the meteorite forced scientists to confront a possibility they had long debated. If the material was truly extraterrestrial, then life was not limited to Earth. It suggested that biology, in some form, could exist elsewhere in the universe. This realization carried implications that reached far beyond the laboratory walls. Astrobiologists began by re-examining old theories. For decades, the concept of panspermia had been proposed, the idea that microscopic life, or its building blocks, could travel across space inside rocks, comets, or dust. The meteorite's genetic material gave new weight to this theory. If structured sequences could survive intact for millions of years, then perhaps life had been moving across planetary systems far longer than anyone had believed. For planetary scientists, the discovery influenced their approach to exploration. The search for life had mostly focused on water-rich environments such as Mars, Europa, and Enceladus. Now, attention shifted to the possibility that genetic systems could survive in unexpected places. If protective molecular coatings and alternative sugar backbones allowed resilience, then organisms might not require Earth-like conditions to endure. Planets with high radiation or thin atmospheres could still support biology built on unfamiliar chemistry. Philosophers of science also entered the discussion. They pointed out that humanity's definition of life was based entirely on local examples: plants, animals, fungi, and microbes that share a common code. The sequences in the meteorite forced a redefinition. Life might not be a single form tied to one planet. Instead, it could be a universal principle expressed in countless ways, some similar to DNA, others completely foreign. Religious and cultural groups followed the news closely. In some communities, the discovery was seen as confirmation that creation was far more expansive than previously imagined. In others, it raised difficult questions about humanity's place in the cosmos. The idea that Earth was unique had guided thought for centuries, and now evidence suggested otherwise. Scholars began to write about how belief systems would need to adapt to a universe where life might be widespread. In practical terms, the implications reached technology and medicine. Genetic engineers studied the alien code for potential applications. If the sequences held methods of protection against radiation, they could be used to design more resilient crops or medical treatments. The unique, triple-stranded structures inspired synthetic biologists, who wondered if similar designs could be applied in artificial DNA for storing data. Suddenly, what had been theoretical curiosity turned into a field with potential practical use. For space agencies, the discovery brought urgency to missions already planned. Mars rovers, Europa landers, and telescope arrays were re-evaluated. Instruments capable of detecting unfamiliar genetic systems were prioritized. If life could take different forms, then the search could not rely only on Earth-based signatures. Teams began designing detectors that could recognize patterns of order and complexity, even if the chemistry was unusual. The implications also stretched into history. Some researchers asked whether Earth's own life might have been seeded by similar material long ago. If meteorites had carried DNA-like molecules across space, perhaps the origin of biology on Earth was not isolated, but part of a larger process. In this view, life was not born on one planet. It was shared, exchanged, and scattered through the galaxy over immense timescales. This idea changed how scientists spoke about humanity's uniqueness. For generations, humans had seen themselves as separate, living on the only world known to carry life. But now, the meteorite suggested that life might be common, with Earth as only one participant in a larger biological network spread across stars. Such a possibility raised difficult questions. If other planets had their own systems of life, what would contact look like? Would humans ever recognize organisms with genetic codes built on unfamiliar chemistry? If extraterrestrial DNA existed, then exploring other worlds carried risks. Missions would have to ensure they did not contaminate environments where fragile ecosystems could exist. International space law, which had focused on resource rights and orbital activity, now had to consider biological protection on a cosmic scale. The discovery also influenced how young scientists saw their future. Students entering fields of biology, chemistry, and astronomy realized they were stepping into a new era. The meteorite had not only expanded science, it had expanded imagination. By revealing genetic material unconnected to Earth, it forced humanity to see itself as part of something larger. Whether that larger reality held microbes, complex organisms, or entire civilizations was unknown, but the discovery confirmed that life, in whatever form it takes, may not be confined to this planet. Challenges and concerns. When the discovery of strange DNA inside the meteorite was first announced, not everyone in the scientific world agreed with the results. Arguments began almost immediately. Some researchers accepted the findings with excitement, while others pushed back with strong criticism. The debate became as important as the discovery itself, because it showed how much was at stake. The first challenge came from contamination concerns. Critics argued that the DNA might not be from space at all, but from Earth. They pointed out that even the cleanest laboratories could accidentally introduce bacteria or microscopic fragments during testing. Some said the meteorite could have been contaminated the moment it landed on the ground, touched by air, soil, or even human hands. These questions forced the original team to release every detail of their methods, from how they collected the fragments to how the samples were sealed and stored. The debate grew louder when new laboratories attempted to replicate the study. Some confirmed unusual results, though not always identical. Others found nothing extraordinary, arguing that the supposed alien DNA was simply rare Earth-based material or laboratory error. These mixed results fueled tension as scientists who supported the discovery accused others of being too rigid, unwilling to accept new possibilities. Skeptics accused the supporters of rushing for fame without enough caution. Academic rivalry also played a role. Some researchers felt excluded from the original project and believed they could have handled the study better. Disagreements were not only about data, but also about recognition, patents, and credit. A few universities pushed their teams to conduct independent investigations, hoping to gain attention with their own findings. This competitive environment sometimes slowed cooperation, as groups worked in isolation rather than sharing results openly. Cultural and philosophical questions also entered the conversation. Religious groups and philosophical thinkers asked what it meant if the DNA was truly alien. Did it suggest that life existed elsewhere in the universe? Did it change how humans understood their own origins? These questions reached beyond science and into society, adding pressure on researchers to speak carefully about what they found. Although most scientists avoided these larger questions, preferring to stay focused on laboratory details, the sequences found in the meteorite did not behave like normal Earth DNA, which made them hard to study. Traditional enzymes and chemical tools sometimes failed to interact with the material. This forced laboratories to design new techniques from scratch, slowing progress. Some tests even destroyed small portions of the sample without producing useful data, which frustrated teams since the meteorite fragments were limited and precious. Another challenge was communication. Scientific language was complex, filled with technical terms. When findings were shared with the public, confusion often followed. Misinterpretations spread quickly, with some claiming the discovery proved aliens already visited Earth, while others dismissed it as a hoax. The gap between cautious scientific statements and sensational headlines created misunderstanding. Because of this, researchers had to balance openness with careful wording. Despite the disagreements, the debates themselves pushed science forward. Questions about contamination led to stricter sample handling rules. Concerns about sequencing errors inspired better machines and improved software. Skeptics forced supporters to repeat experiments under more controlled conditions, strengthening the overall reliability of results. Even rivalries produced progress, as competing teams raced to refine methods. International collaboration faced difficulties too. Some countries demanded access to the samples, arguing that a discovery of this scale belonged to humanity, not just to one group or nation. Others insisted that ownership should remain with the institutions that recovered the meteorite. This dispute over access slowed cooperation, as negotiations over rights and responsibilities continued in the background. The debates highlighted the fragile nature of groundbreaking science. When discoveries challenged established knowledge, resistance was natural. The conflict was not simply about data, but about human pride, ambition, and the struggle between tradition and change. If the strange DNA had been ignored or accepted too quickly, the risk of error would have been enormous. Instead, every claim was dissected, every detail questioned. The process was messy, sometimes emotional, but it was also evidence of science at work, future research, and exploration. After the discovery of the unusual sequences inside the meteorite, attention quickly shifted to the future. The scientific community understood that one rock, no matter how extraordinary, was not enough to rewrite biology or reshape human understanding of the universe. More samples were needed, more missions had to be planned, and new technologies would have to be built. The next steps became the focus of endless discussions across the world. One of the first priorities was preservation. The meteorite fragments already studied were placed in deep freeze storage at specialized facilities. Only a small portion had been used for testing, and the rest was kept for future methods that might not yet exist. Scientists understood that technology evolves quickly, and what could not be analyzed today might become clear with tomorrow's instruments. The samples became reference points, benchmarks for all further research. Parallel to preservation, funding bodies began drafting proposals for targeted collection missions. If one meteorite had carried such material, then others might as well. Projects were drawn up to expand recovery networks on Earth. High-altitude balloons and specialized radar arrays were suggested to track incoming meteors more precisely, allowing faster recovery before contamination could occur. Desert regions, polar ice fields, and ocean floors were identified as prime locations for searching. Space agencies turned their focus outward. Missions already planned to Mars, Europa, and Enceladus were reviewed and expanded. Instruments capable of detecting complex organic sequences were added to payload lists. Engineers began designing portable sequencing devices that could operate in space without contamination from Earth. These tools would allow astronauts or robotic probes to test samples directly on alien surfaces, reducing the risk of transporting fragile material back to Earth. Beyond hardware, attention turned to collaboration. No single country could carry the burden of studying extraterrestrial genetic material. International consortia were formed, linking institutions across continents. Shared databases were created to log every sequence, every test, every observation. These platforms allowed researchers to compare results in real time, reducing duplication and increasing trust. Transparency became a priority since skepticism was still strong within parts of the community. Ethical frameworks were also drafted. If future missions encountered intact organisms, how should they be handled? Could they be returned to Earth, or would they have to be studied in off-world labs? The risk of cross-contamination went both ways. Earth microbes could threaten alien ecosystems, and alien systems could potentially interact with terrestrial biology in unpredictable ways. International committees debated protocols, aiming to set boundaries before the next discovery arrived. Meanwhile, geneticists and chemists pressed forward with laboratory work. They built synthetic copies of the unusual sequences, not from the meteorite itself, but from digital data. This allowed them to test how the code functioned without risking the original samples. Some of these synthetic strands behaved in unexpected ways, forming stable structures under conditions that normally degraded Earth DNA. These experiments opened new avenues for biotechnology. Astronomers also joined the effort. Telescopes were directed toward regions rich in comets and asteroids, scanning for potential targets that could hold organic material. Missions to retrieve pristine samples from these bodies gained priority. Projects, like sample return capsules from distant asteroids, were now seen not only as geological missions, but as biological ones. The question was no longer just what minerals they carried, but what genetic codes might be hidden inside. Private companies also saw opportunity. Firms specializing in space technology offered to develop sequencing tools, clean lab environments, and advanced storage systems. Biotechnology companies explored partnerships, curious about what lessons could be learned from alien molecular stability. For the first time, extraterrestrial genetics was not just a scientific subject, but a potential industry. At the global level, the discovery influenced long-term planning for exploration. Discussions about crewed missions to Mars included new questions. If astronauts found genetic material there, how would they study it safely? Spacecraft designs were reconsidered, with quarantine sections proposed for samples and stricter sterilization of outbound equipment to avoid contaminating other worlds. The path forward was clear but challenging. More meteorites would have to be studied, missions beyond Earth would need stronger instruments, and humanity would have to prepare for the possibility that the strange sequences were not isolated curiosities but part of a much larger story written across the galaxy. The discovery had set the direction. The future now belonged to exploration guided by a simple goal: to follow the trail of life wherever it might be found. What are your thoughts on the possibility of life existing somewhere else in the universe? Let us know your views in the comments. If you enjoyed this video, like and subscribe to this channel. Before you go, watch the other videos on your screen.